Recording it.
Okay. Now, in immunity, I'll go straight to what I think is confusing you and what is important, which is the types or the immune responses. Yeah, I'll start from here because you have a problem with this part. Now, what's the difference between the primary and the secondary immune response? There is a big difference between the primary and the secondary immune response. Primary immune response, from the name, you have an antigen that will enter the body for the first time. And also it includes vaccination. Noor, remove your phone and remove your bag. Okay, so infections with the first time or for the first time. Primary immune response means what? Infection for the first time with any antigen, with any disease, or you took a primary or an initial vaccination dose. And this is going to happen if any foreign antigen enters the body. Okay, the first thing that will happen is that the antigen will enter the body and the body is going to inflate. There will be inflammation, an increase in the blood flow. So you will start with the inflammation. The inflammation happens because either the body's own cells produces a chemical called histamine. Histamine is the response of an antigen, a foreign antigen, or due to the entrance of the foreign antigen itself, it's going to cause inflammation. This inflammation can be external, you can see it, or can be internal inside your body. In all cases, you have an inflow of blood. Okay, so you have inflammation, either due to the release of the histamine, or it is due to the foreign antigen itself. It's going to cause vasodilatation, increase in the blood flow. Increase in the blood flow. What will come with the blood flow? Which cells? The first line of the set of defense. Yes, the white blood cells. Which exactly? Phagocytes or lymphocytes? Phagocytes. So the second thing you will mention is that the white blood cells or the phagocytes specifically will arrive and they will do what? Phagocytosis. And you have a whole list of the steps of phagocytosis. You have the direct attachment, or they will attach because they are marked by antibodies, extension of the cell membrane, causing a phagosome. The lysosome will arrive. Lysosome plus phagosome will fuse together to cause a what or produce a what? Phagolysosome. Hydrolytic enzymes will be released. Mention one or two examples, and then digestion and killing. Okay? Now, in the phagocytes, you studied two types of phagocytes. You studied what and what? Neutrophils and macrophages. Very good. The neutrophils, they will do the complete phagocytosis. They will completely break down the antigen. And the macrophages, they do it a little bit differently. Can you remind me, Hedy? Okay. Very good. And they turn into a very good. So they do incomplete phagocytosis. They leave part of the antigen. They combine it with a major histocompatibility complex, and then they present it on the surface of the cell, and it will become an antigen presenting cell. And this is a very important step. This antigen presenting cell will ask for help. So it will activate which cell? T helper. And the T helper will release a substance called what, Yasin? Cytokine. And the cytokine is going to wake up the whole immune system. What does the cytokine do, Noor? It will activate the second line of defense, the other white blood cells, which are what? The lymphocytes. Yes, the cytokines will activate the lymphocytes, or they will activate more angry macrophages. That's it. So it is very, very simple. Let's break it down. We have here a foreign antigen. Fine, it will enter. Now, this is the pathogen, whether a bacteria or a fungi or a virus or whatever. It has a molecular marker on its surface, which is a protein called the antigen. And this antigen is not from inside the body, it's self or non-self? Non-self antigen. It will activate the immune system. Number one, I have vasodilation. So I will start with inflammation. I will mention that there is inflammation. I have a flow of blood. With this flow of blood, I will have the phagocytes, they will arrive. These phagocytes are either neutrophils or macrophages. In all cases, you will mention phagocytosis and you can briefly describe the steps. Macrophages here, they will turn into antigen-presenting cells. They will take part of the antigen and they will present it. When they present it, they will activate the T helper that will release the cytokines that will activate the lymphocytes, whether the B or the T lymphocytes, and they will activate more macrophages. Until here, is everything clear? Okay, Mariam? Good. Now, you have a problem after the B lymphocytes are activated. I don't know why. Now, whether the B lymphocytes or the T lymphocytes are activated, so we have, again, the activation of the T helper, the release of cytokines, and then cytokines will activate the lymphocytes, whether the B lymphocytes or the T lymphocytes. And then here you mention the colonal selection and the colonal expansion. Because will I activate all the B lymphocytes in your body? Will I activate all, Noor, I'll take your phone. Okay, pay attention. Will I activate all the T lymphocytes in your body? No, only one. Only the one specific lymphocyte that has a complementary receptor that will bind with the antigen. Okay, that's it. So here you will mention colonal selection and just you will mention a sentence that a specific B or T lymphocyte will be selected that has a complementary receptor, complementary to the antigen. Finished. And then colonal expansion. What do you say in colonal expansion, Adham? Whether B or T, this happens in both. Very good. Exactly. The selected lymphocyte, whether the B or the T lymphocyte, will amplify, will increase in number, will create an army by multiplying by mitosis. Good. And then they will differentiate. This is the part that confuses you, and I rewrote it again in a better way, and I sent you the PDF. Okay, they will differentiate. Now, again, you are confused here. When the B lymphocytes differentiate, B lymphocytes, they differentiate to produce what? Very good. When the B lymphocyte differentiate, they differentiate to give you two things. Plasma cells, or they remain in the blood and they become memory cells. And the plasma cells, what is their function? They produce what? Antibodies. That's it. So the B lymphocytes or the plasma cells are the only cells that produce antibodies. Now the T lymphocytes, here is the differentiation. The B lymphocytes will either give you plasma cells or memory cells. And the T lymphocytes, here's the nice B lymphocyte. It will differentiate plasma cell that will give me antibodies or memory cells. Good. When the antibody is attached to the B lymphocyte itself or the B cell, it's called the B cell receptor. When it is released into the blood, it is called the antibody. Fine. Where am I? I have nice pictures, but I like to do my own drawings. Now the T lymphocyte. Again, the T lymphocyte will go through the same process. If it is activated, then I will choose only one T lymphocyte that has a complementary receptor. This is coronal selection. And then I will select one. And then this is going to keep on dividing so that I have many of them by mitosis. And then the T lymphocyte will differentiate. They will differentiate to give me what? What are the types of T lymphocytes? T killer or T helper or memory. Okay, so this T lymphocyte will differentiate either to give me more T helper, and you know its function. This is the one that releases the cytokines and wakes up the immune system, or the T killer. Of course, the T killer here is the most important because the T killer is what we want to kill the pathogen, or it will remain in the blood to give you memory cells, memory T cells. That's it. This is the part that confuses you. Okay, so again, and they keep on more or less asking you over this part. So from the very beginning, I have a primary infection, I have an infection, or I took a vaccination dose. Now this vaccine is a pathogen, so you know it is weak or it is dead, whatever. This entrance of the antigen is going to activate your immune system. Number one, what do you have? Inflammation. This inflammation will cause an influx or flow of blood. This inflammation will bring with it what? Phagocytes. You will mention phagocytosis here. Discuss it in brief. Phagocytosis is done by two types of white blood cells. What are they, Hedy? Neutrophils and macrophages. These macrophages, they will, what, Farouk? They will turn into antigen presenting cells. So you have to mention in your answer that you have antigen presentation. This antigen presentation or the antigen presenting cell will activate the T helper. Don't get confused also. Some students, they write the cytokine before the T helper. Okay? Think about it this way. When the macrophage turns into an antigen presenting cell, it will ask for help. So it will activate the T helper. Okay, no? Very good. That will secrete the cytokine. That will activate the second type of white blood cells, which are the lymphocytes. From here, the confusion starts. Okay? Tell me, Adham, 1 minute. Lymphocytes are now activated. They will go through what? Both of them, whether B or T. Clonal selection. I will select one, the one with the complementary receptor. And then, very good, clonal expansion. They will multiply by mitosis and then differentiation. And I'll be done. Okay? It depends on which B lymphocyte has been selected. If it's the B lymphocyte, it will differentiate into what and what? Hedy? Plasma, that will give me antibodies, and memory that will remain in the blood. T lymphocyte, Kenzie, will differentiate into? Okay, if they remain in the blood, they will. T cells never produce antibodies. Either T helper or T cytotoxic. Okay? Antibodies are only produced by the plasma cells, only B lymphocytes. So either the T lymphocytes, when they're activated, either they will give you T helper or T cytotoxic. Now, you know the function of the T helper very well. The T cytotoxic or the T killer cells, they're part of the cell-mediated immunity. Cell-mediated means they will attack the cells directly. Okay? They will attack the infected cell, usually by releasing hydrogen peroxide or something called perforin that will make perforations or holes in the cell wall of the pathogen so that the water will enter by osmosis and the turgid pressure will increase inside this pathogen and it will burst. Okay, fine. Now also this is another part or another point of confusion. The B lymphocyte, my dear, B lymphocyte Kenzie, is the cell that will differentiate into the plasma cell, and this is the cell that will produce the antibody. T lymphocytes do not produce antibodies. No antibodies. T lymphocytes, if we are talking about the T killer cells, they directly kill, they attack. These are cell-mediated cells or cell-mediated immunity. The plasma cell and the antibodies, they work in the bloodstream. So this is known as the humoral immunity or blood-mediated inside the blood. Okay, and this is sometimes one of the reasons why the antibody cannot kill the pathogen, because the pathogen is hiding inside the cell and the antibody is in the blood. Okay, this is half the lesson. This part is half the lesson. Good. Now let's go back to the PowerPoint so that I don't rush. So picture by picture, I hope it sticks. Okay, so you have clonal selection, clonal expansion, differentiation. Those three steps happens whether it is B lymphocyte or T lymphocyte. But the difference is that every one of them differentiates into something else. The B lymphocyte plasma and the T lymphocyte T helper or T cytotoxic. Easy? Okay, we're done with that. Now a little bit, some information about the B and the T lymphocyte. The T lymphocytes, they are produced in the bone marrow, but they mature in the thymus gland. Here, the thymus gland. It's here. The B lymphocyte, it's easy. It is produced in the bone marrow and it matures also in the bone marrow. Okay? Maybe they might ask you in paper one. Now, this is the primary immune response. We finished it. When you understand it and you study it as a story, it gets very easy. Is it clear? Inflammation. With the inflammation, I have the neutrophils, phagocytosis, and macrophages, antigen presentation, and then clonal selection, clonal expansion, differentiation. Finished. So after the primary immune response, Hedi, what will remain in the blood, Noor? After the antibodies are produced and they kill the antigen and some time passes, what remains in the blood? Memory cells. And the moment you're asked about the secondary immune response, secondary immune response, okay? Secondary means what? You are infected with the same pathogen, reinfection with the same pathogen. This is when you start with the word memory cells, because memory cells remain from the primary immune response. Okay, so you will start with the word memory cells. Memory cells that remained in the blood, they will quickly detect this foreign antigen, quickly, Hedi, divide by mitosis, increase in number, and quickly differentiate into plasma cells if they were B lymphocytes, or T cytotoxic if they were memory T cells, and quickly take action, so that you are better without even becoming sick. You're immune. The virus entered your body and you don't know because you did not show any signs and symptoms. Okay, so Noor, when you are asked about the secondary immune response or the immunological response, the first word you will start with is the word what, Hedi? Memory cells that are already in the blood. They will quickly become activated, quickly divide by mitosis, quickly differentiate, quickly either produce antibodies if they are plasma, or they will directly kill the cell if they are T cytotoxic. Okay, that is why this secondary immune response, Adham, if you are asked to compare this one with the primary in terms of strength and speed and duration, what will you say about the secondary immune response? Faster response, faster production of antibodies if it is in the case of the B lymphocyte. And the response is weaker or stronger? Stronger, okay? That is why you have this graph here. This graph, I think you saw it a lot. You have the time on the x-axis and the antibody concentration on the y-axis. The first part of the graph, this is the initial infection, the initial entrance, because if you noticed, a lot of things happen until the antibodies are produced. First, I have inflammation, and then phagocytosis, and then antigen presentation, and then clonal selection, and clonal expansion, and then differentiation. So this takes a few days. Maybe it will take a week before antibodies are produced. Okay, Mariam? And then after everything is finished, we have a war here. After it is finished, I have memory cells. I have memory cells here, they remain. Once there is a secondary reinfection, and you have to say that we have been exposed to the same antigen, quickly the memory cells here will take action. Quickly, they will differentiate and divide and produce the antibodies. That is why the secondary immune response is faster and stronger. And you will not even show signs and symptoms. The bacteria or the pathogen will be dead without even showing signs and symptoms. So that is why sometimes they can ask you, why do you take a booster dose? So that you boost the memory cells to make the response of the immune system stronger and faster. Always use these two words, okay? To activate the memory cells, to make sure that they're there, to make the immune system stronger and faster, to produce more memory cells in the blood. Okay? Is this part clear now? Clear? Okay. So here, the primary immune response, clonal selection and clonal expansion. Clonal selection and expansion is in the primary, not the secondary. The secondary, only you start with the memory cells. Memory cells will take action and their action will be very quick and very strong. Is this now clear? This graph, is it clear? Okay. Here in the primary immune response, actually, you say a lot of things. You say the 4 steps: inflammation, phagocytosis, antigen presentation, clonal selection, clonal expansion, differentiation. Okay. Secondary immune response, you will only talk about the memory cells. That's it. And how quick and fast they are and blah, blah, blah. Okay, Yassine? Is it easy now? Okay, good. So we will not go over that. Now, also, I definitely got this from a question. How does the plasma cell produce antibodies? What is the nature of the antibody kinesin? It's a protein. So the moment they ask you this question, you will write all the steps of protein synthesis: transcription and post-transcriptional modification, translation and post-translational modification, and we revised it. And then the modification in the rough endoplasmic reticulum and the Golgi body. And please give examples whenever possible. Okay? Okay, Farouk? Easy? Immunity. How? Okay, antibodies are produced. How do they go out? This is another lesson. Exocytosis. Mention the exocytosis. It's a fusion with the cell membrane release. So easy, it needs energy. So I think now this is clear. Okay, Heidi? Good. Exocytosis. I think the picture speaks for itself. So this is a common here exam question that we already asked, that we already answered, and this is in your notes, so you'll find it. So how the antibody response following a second infection will differ from the primary infection: faster, quicker, higher concentration of antibodies, and that's it. These are the main points. Faster. This was a question out of 3 marks. Of course, you mentioned the term memory cells. Okay. Is this part done? Clear now? Okay, let's go back to the very beginning. Because do I give you a break? Huh? Yeah, 1 minute. Okay. Let's start with the antibodies. The structure is very important and very easy. Please remove your phones. Okay, so when you come to study the structure of the antibody, Yasin, you will immediately draw it. Okay, you will immediately draw it. It's very easy. Now, what is the nature of the antibody? It's definitely a protein. What will you draw? You will draw this. 2 heavy chains, Noor, okay, Y-shaped, and 2 lighter chains. And Noor, you will help me label. And then I'll change color, Noor, and I'm going to do this. Okay, can you tell me the nature of this protein? Is it a primary or a secondary or tertiary or a quaternary protein? It's made up of one polypeptide chain or more than one polypeptide chain working together. More than one polypeptide chain working together. So this is a quaternary protein. More than one polypeptide chain working together as one unit. So is this a fibrous or a globular protein? Globular. Globular means it is water soluble because the hydrophilic R groups are on the outside. Nearly spherical. This is not very spherical. Okay, globular, nearly spherical, soluble in water. And when you are asked to discuss why is it soluble in water, R groups are positioned on the outside and the hydrophobic on the inside. Now, when the hydrophilic R groups are on the outside, this creates what type of bonds with water, Yassine? Hydrogen bonds with water. Don't forget the solubility part. They always ask about it. Okay, so it's a quaternary protein. It's globular, soluble in water. It is Y-shaped, as you can see. It is very obvious. Okay, now let's talk about the different parts of this globular protein. These two chains, the longer ones, what are they called, Noor? Heavy or light? Heavy. Can you study? They're called the heavy chains. And as you can see, you have two heavy chains, 1 and 2. The heavy are the longer chains, those. These are the heavy chains. Okay? Fine. Now, they're gone. Okay, now the shorter chains, what are they called, Hedi? The shorter chains? The light chains. Good. Now, what is this part, Adham? You are talking about these? Yes, these are the disulfide bridges that hold the heavy and the light chains together. They're also found here. Now, this orange part, what is it? It's called the hinge region. And what does it provide? Flexibility. Is it found in all antibodies or not? No, it's not found in all antibodies. Some antibodies, they have the hinge region and some don't. Okay, provides flexibility, makes it move, set itself at certain angles while it binds with the antigen. Fine. Now, this part, the part that binds with the antigen, Hedi, what is it called? Yes, this small part is the variable region, and it is called variable. Why is it called variable, Mariam? Yes, it's specific. It has a certain sequence of amino acids. We did solve, by the way, several questions, and they ask you about the variable region. And when you come to describe the variable region, you have to mention specific, binds with a specific antigen, and it has a specific sequence of amino acids. Okay, how many variable regions do you have, Noor? No, 4. Can't you see? 1, 2, 3, 4. This whole part, the part that binds with the antigen, is called the antigen antibody binding site. And it binds with a specific part on the antigen called the epitope. Okay, so we broke it down. This is a globular protein soluble in water because the hydrophilic R groups are on the external area towards the water, hydrophobic inside. The hydrophilic R groups, they form hydrogen bonds with water, quaternary, made of more than one polypeptide chain working together as one unit. Now, this part is called the constant or the variable region, constant region, and it determines the type of the antibody. How many types of antibodies do we have? 5. Megid, remember? M-A, whatever, G-E-D. And every type or class of antibody, they have a specific mode of action because antibodies, they work differently. And we will discuss this in a minute. Now, the ends of the heavy chains, these are the parts that bind with the receptor on the phagocyte. Remember the phagocyte? The phagocyte will bind with the antigen if it will bind with the antigen, yes, if the antigen is marked with a specific antibody. Remember phagocytosis? In phagocytosis, what is going on? Okay. I don't want that. Okay. In phagocytosis, we have the phagocyte or the neutrophil. It will attack the pathogen either directly or the pathogen itself is marked with an antibody, and the phagocyte will have a complementary receptor. This is one of the methods of the antibody production. marking the antigen. Okay, Kenzie? Now, what are we talking about? Yes, we're talking about the constant region. This constant region, it binds with the complementary receptor on the surface of the phagocyte. So what is the function of the constant region? If they ask you, the constant region is made up of the ends of the two heavy chains, and it determines the class of the antibody, and it binds with the complementary receptor on the phagocyte. Good. Now, how does the constant region determine the type of the antibody? What are the different modes of action of the antibody? We said, how will we remember them? How does the antibody operate? We said there is a sentence that we use to remind us, how does the antibody kill the antigen? I know ball. Yes, I know ball. So this is done. This is the constant region. This is the variable region, antigen antibody binding site. Done. Okay. Here, we said I know ball. What is I? Let's look at the pictures. What's I, Hadi? I will remind you with which word? Immobilization means it will make the antigen or the pathogen stop moving. Okay? This is immobilization. It will attach to its flagella. Now the picture speaks for itself. The antibodies attach to the flagella of the bacteria, make the bacteria immobile. They cannot move, so they cannot spread. Okay? I know ball. What's N? Neutralization. What does it neutralize, Hadi? The toxins released by the pathogen. This is neutralization. What is O? Opsonization, which is marking of the antigen. Here, this is opsonization. Here is my antigen, and it is marked with an antibody, and the phagocyte has a complementary, what is this? Receptor. This is the receptor on the phagocyte. It is complementary to the antibody so that I can mark it. I can see it and I will go and swallow it. Okay? Now this is opsonization. What's B? Block. I will block. No, you will not attack my host cell. This is blocking. What is A? Agglutination. It will make the pathogen stick together or clump together. Okay, Noor? Okay, what is L? Lysis. It will make holes in the cell wall of the pathogen. Water will enter by osmosis, increase in the turgid pressure. It will burst. Very easy. So antibody. So these are the two important parts of the lesson. The immune response, whether the primary or the secondary. And here I have the antibody structure and function. To study it, you will draw the structure and label it. And you're done with the structure. Function, I know more. Finish. Okay, good. Noor, any questions? Fine. What do I want to say here? That any interactions between two proteins is always done with the R groups or the side chains. So we said all that, and these are just more pictures. Okay, here is the marking or the opsonization. Here is the immobilization. Here is the clumping and the lysis. And you have all these pictures to look at. Now, making monoclonal antibodies. Very important and also very easy. This is a nicer picture. Farouk, can you describe this? What did I do to this mouse? I will inject it with a what? Okay, yeah, a foreign antigen or a pathogen. And what will this do inside its body? Very good. Yes. Very good. Wait, stop here. Why do we have to combine the plasma cell with the tumor cell? In the plasma or the tumor? In the tumor cell. Okay, the tumor cell is immortal. It doesn't die. No apoptosis, uncontrollable mitosis, and we want this feature. So we will combine both together to give me what? Hybridoma cell. And then the hybridoma cell, I'll clone it so that it gives me the monoclonal antibody. We use, now I don't want you to get confused. This is an antibody, not a pathogen. This is an antibody. We are creating a monoclonal antibody, not a pathogen. So we will use it in which type of vaccination, Noor? Active or passive vaccination? Passive. Okay, we will discuss the two types of vaccination in a minute. So this is another important objective. I will generate monoclonal antibody. Why did they call it monoclonal antibody? Monoclonal means one antibody. I will make artificially in the lab one type of antibody to attack a specific antigen. Okay, now if I want to create a monoclonal antibody against a specific antigen, I will inject this specific antigen into the body of a small mammal, like a mouse. I will activate the primary immune response, everything that we said, inflammation and phagocytosis and antigen presentation, clonal selection, clonal expansion, differentiation, everything will happen in the body of the mouse because I want to extract from the mouse the plasma cells that will produce this specific antibody. I do not extract from the mouse the antibody. No, I extract the plasma cell that will give me the antibody. This plasma cell has a problem. It will die very soon. So I will have to fuse it with a tumor cell, myeloma cell or tumor cell that does not die. When I fuse them together, how do I fuse them together? I use what? Fusogens. And give me an example of a fusogen. Fusogen is something that will fuse the plasma with the tumor cell. Yes, a small current called electrofusion or a chemical known as polyethylene glycol or just PEG. Okay? Okay, Farouk? Good. I'll fuse them together. This will give me a hybrid cell or a hybridoma cell. And this hybrid cell, there is one step here, humanization, because this hybridoma cell came from a mouse, so it can cause an allergic reaction. I will do some humanization. First, I will screen it to make sure that it is giving me the antibody that I want. And then I will humanize it. I will change the sequence of amino acids to make it more human so that it does not cause an allergic reaction more. And then after I am done and satisfied, I'm going to clone it. I will clone the hybridoma cell, okay? And this will produce lots and lots of antibodies. And now I can use it in a lot of fields in medicine. Diagnosis and treatment and blah, blah, blah. Okay. Now, the idea about the monoclonal antibody, this is my antibody. This is how we draw it in short when we come to draw it. This monoclonal antibody, Farouk, I can attach it to anything, to an enzyme, to a drug, to a fluorescent label, anything. Okay. Because this monoclonal antibody will go and bind to a specific antigen. Once I inject it into the blood, it will go and bind to a specific antigen and deliver this drug or mark the antigen or take the enzyme with it to catalyze a particular reaction, so on and so on. Yassine, why don't I take this monoclonal antibody by mouth? Yes, Adham. It will be digested, yes, where? In the stomach, because in the stomach we have the pepsin enzyme, and the small intestine we have the trypsin enzyme. So it will be digested and it will be useless. I don't want it to go into the digestive system. Immediately I will inject it into the circulatory system. Good. Now, from there, you will understand that the monoclonal antibody is used in diagnosis. It is used in treatment. It is used in localization of infection, anything. I will attach the monoclonal antibody to anything. If it is attached to a drug, then this is called localized therapy, localized treatment. This monoclonal antibody will be attached to the medicine, enter your body, and go attached to the disease cell. And this is usually used in cancer to spare the healthy cells. Okay, I will use it for diagnosis, mainly diagnosis, because I'm going to attach the monoclonal antibody to a fluorescent marker, something that will shine under a screen. I have lots of pictures. I don't know why I don't use them. We talked about the humanization. Here, the disulfide bonds. This is the antigen antibody site. These are the variable regions. We discussed it. And we are at the uses. Uses, diagnosis, treatment, food safety testing, pregnancy tests. Good. Now I want to use, when we will attach it to a fluorescent marker. Here is this is the antibody. It is attached to a fluorescent marker. I will inject it to the blood. It will bind to the antigen. And then I'll put the person or the human being under a machine. Okay. And this machine is going to strike some sort of radiation so that the fluorescent marker will shine. And I will see the location of the antigen inside the body. This is the machine on a screen. I have a screen. Okay. And I have the patient mapped on the screen. This is my patient. He's sleeping, and I can see the fluorescence here. So, Yassine, I can know that the thrombus or the cancer or the infection is here and how big it is and the prognosis, the degree of where am I if I am in a certain stage in cancer, even the type of infection. So it's very accurate, it's very quick, it's very precise, it cannot do mistakes because the monoclonal antibody will bind with a specific antigen. Nice? Anything hard about it? So antibodies, again, when I come to study it, I will draw it to draw the structure. I'll break it down, the two heavy chains, the dual light chains, the disulfide bonds, the hinge region, and the uses of the hinge region, the variable region, and the antigen antibody binding site. And here it binds with a complementary receptor. So I'm done with the structure. How does it work? I know ball. So I don't forget, believe me, this really helps you. Immobilization, utilization, opsonization, blocking, agglutination, lysis. This is the function. And then uses. This monoclonal antibody, I bind it with whatever I want. And I can even not bind it to anything. I will use it in passive immunity to go and kill the antigen. To take. Mm. What if it doesn't? What if we want to send a particular drug that has a different mode of action? Say cancer. If cancer was killed by antibodies, then no one would die from cancer. Yeah. It will only be used for diagnosis. Yes, this can show that we have been treated. Okay, so that's why it helps us in knowing the stage of the treatment. We are getting better, we're getting worse. Yeah. Oh. Okay, or we cannot bind anything, just the monoclonal antibody. In this case, we will use it in passive immunity. So it has lots of uses. Passive immunity, localized treatment, diagnosis, to know the exact type of infection, the location. We use it in specific examples in your syllabus to know the location of a deep venous thrombosis. You have a thrombus in the leg. Usually it's deeply situated in the leg. We want to know where exactly. We want to know where exactly. So we will inject an antigen. What is the protein that is found in blood clots? Fibrin. Remember in year 10 when we took that, soluble fibrinogen will be converted to insoluble fibrin that will create a mesh and attract circulating red blood cells and white blood cells to make all that. Remember all that? So this blood clot contains fibrin. So we will create a monoclonal antibody. We will inject our mouse with the fibrin and it will activate its immune response and create plasma cells to produce antibodies against the fibrin. I will extract this plasma cell, fuse it with a memory cell, create a hybridoma cell so that I have a monoclonal antibody. I will inject this monoclonal antibody into the patient's body so it will go directly and bind to the fibrin. I'll know the location and the size of the fibrin. Okay, Noor? Done? Enough? Not enough. I mean, is it clear? Okay, it's a long lesson. Every single part in it is important, actually. 1 minute break, the minute is finished. Okay, the types of immunity. We took this last year, it's O level, so it should take me a minute. Okay, Farouk? We have active, we have passive. Now from the name, I will not ask questions, I'll finish. Active immunity, from the name, the immune system is activated. I have antibodies that will be produced. I have memory cells that will be produced. Okay, this is active immunity. If this happens because I got a natural infection, you see, this is natural or artificial active? Natural. If I will take a vaccine, artificial, finished. Okay, passive immunity. From the name, the immunity is sleeping. No antibodies produced. No memory cells are produced. The immunity is in la la land. It is sleeping. I did not activate the immunity. I took ready-made antibodies that will do the job and then they will be broken down after some time. Either I take them from my mother or I will take them in the emergency room because I was bit by a dog. Okay, you were never bit by a dog before? It's a very nice experience. Good? Seriously, you never bit by a dog or a cat or anything? No? Seriously? And then? Good. That was a street dog or someone else's dog? Oh, the rabies, yeah. It happens. No, I was scratched badly by a cat. Okay, and I took the monoclonal. That was actually recent. Yeah. House cat and a superficial scratch or a deep one? A superficial scratch or a deep scratch? It makes a difference. Yeah, okay. Oh God. Especially street. I was scratched by a cat. I stepped on a cat. She was sleeping and she was a baby cat and she bit me on my leg. Yeah, that was actually at the beginning of this year. I didn't see it. It was too dark and she was sleeping under a plastic bag and I stepped on it. So I feel sorry for the cat. She had to defend herself. So it bit me and I went to the ER and I took 5 injections. It's not a nice experience. Okay. So is this easy? Sorry. And then what is we know this very well, which is the primary vaccination dose and the secondary, the same thing as the primary and the secondary immune response. What is new to the AS syllabus are the type of vaccines, not the types of immunity, not the type of vaccine itself. The vaccine, what does it contain? Either a live pathogen or a dead pathogen. This live pathogen, it's alive. The bacteria, it's alive, but it is weak. And this inactivated vaccine, it's not a pathogen. It's dead, or it is the toxin produced by the pathogen called the toxoid. Now, which do you think, Farouk, is stronger? The live. Okay, it's stronger. It gives you a stronger immune response, but it has disadvantages. What can it cause? Yes, it can cause the disease itself if you have poor immunity. Any type of vaccine can cause an allergic reaction. The most disadvantage is that, and the live vaccine is that it can cause the disease itself if you're immunocompromised, such as an HIV. Good. In the inactivated virus, it is very safe. It will never give you the disease because the virus is dead or it's just a toxoid. Okay? And this is very suitable for very little infants, like 2 or 3 months old, or if you have a very weak immune system. Nice. Now, this is important. Disadvantages of active immunity. Now, you know that they love asking you about vaccination. Why would a program succeed? Why would a program fail? Why or what are the advantages or the disadvantages of a vaccine? So this list is important. It needs medical staff. It is expensive. It will not give you an immune response. It will fail. You need a booster dose, means you need more money. You need to store it suitably. It can cause an allergic reaction. If you have a weak immune response or a weak immune system, you will become affected. And the live vaccine means the live pathogen will be released in your feces. And this can cause more spread of infection if the whole population is not vaccinated at once. This is it's a very common question. This together with the infectious diseases. Disadvantages of passive immunity. Expensive, temporary. This is the most important disadvantage, that if you're bitten by a dog again, you will go again and take a passive immunity. You'll take an injection because monoclonal antibodies, they have a certain lifespan in your body and then they are broken down. Okay? If a person is reinfected, they would go and get another monoclonal antibody again, and any one of them can cause an allergic reaction. I think why can't we make a vaccine against HIV? Yes, mutation, antigenic variation, antigenic shift, antigenic concealment. Yeah, I think you know this very well. Okay, it doesn't finish. Antigenic shift means it will mutate even during its life cycle. It's even smarter. Yes, it can variate more than once during its life cycle. Okay, we are done with all the diseases. Sure? The HIV, you know how does it enter the T lymphocyte? It has a glycoprotein receptor that binds with CD4 receptors. Yes, you have to memorize it on the T helper cells. So it will enter the T helper, destroy it, and when it destroys the T helper cell, it destroys your immune system. One, just be quick. You know the self and the non-self. This is so easy. The different types. You know the myasthenia gravis? The myasthenia, this is a neuromuscular junction. This is a nerve cell and this is a muscle. And the impulse normally travels and binds with the receptor on the muscle to make, no, where are you going? We're going to the lab. Okay, making the muscle contract. Okay. Now what happens is that the body creates an antibody that will block the receptor. So the impulse will not bind with the receptor and the muscle will not contract. And you have slow paralysis. This is myasthenia gravis, an autoimmune disease. Okay, easy, be patient. So I don't miss anything. And the multiple sclerosis here, you have also an autoimmune disease that will eat your myelin sheath. The myelin sheath will be broken down. So you have nerve diseases, you have tremors, you cannot focus, you don't have sensation, blah, blah, blah. Okay, multiple sclerosis. Rheumatoid arthritis, the name speaks for itself. You have the immune system attacking your major joints, inflammation in your joints, and moving becomes very difficult. Okay, wait, you know diabetes, it will attack your pancreas. The pancreas will not produce insulin, so you will not be able to regulate your glucose levels, and you will have diabetes. Your glucose levels will be very high. Good. This is what systemic lupus erythematosus, which you have generalized inflammation, and the autoimmune disease attacks your skin. And the leukemia, a type of cancer. Now, in any cancer, you will mention the two genes that will go, proto-oncogenes that will change to oncogene, and the tumor suppressor gene will be switched off. I have uncontrollable mitosis, no programmed cell death, no apoptosis. I have a ball of undifferentiated, unfunctional, useless tumor that only cause trouble, like you can see here. They do not respond to external stimuli. They have their own blood supply. They're not killed by the immune system. They cause cancer. Okay? No, the proto-oncogene is changed into oncogene. Yes, deal quiz. Okay? We have a shortened life cycle, fast replication. I think we took this in protein synthesis also. So, and any type of cancer can be acute or chronic. This is systemic lupus erythematosus. You don't have to know its name. I just want to make sure here, proto-oncogene to oncogene. Yes, we said all that. Yes, the first part of the lesson just that the bone contains the adult stem cells that differ. They can ask you about the stem cells. What are stem cells, differentiated or undifferentiated? They divide by mitosis, either to give you more stem cells or they can differentiate to replace any damaged cell. They repair tissues, but they replace cells. In the bone marrow, you have myeloid or lymphoid stem cells. I'll just say this and let you go. The myeloid will give you the phagocytes. And the lymphoid, from the name, they differentiate into the B lymphocyte and the T lymphocyte. So I don't think we missed anything. And the structure, the neutrophil is irregular with a lobe nuclei. And the macrophage has a kidney-shaped nucleus. It's the biggest. That's why it's called macrophage. And the lymphocyte is circular with a large central nucleus. This is the lymphocyte. I think you know those, and you know phagocytosis very well. And don't forget the enzymes that are found in the lysozyme, hydrolytic enzymes, proteases, neutralases, nucleases that break down the peptidoglycans or the phosphodiester bonds. What is the name of the enzyme that is broken down when we don't want to create crosslinks in the peptidoglycan cell wall in bacteria? We took it in drugs. Transpeptidase enzyme. Okay. Okay. So is everything clear? Okay, good. Very, very good. Let's go to the lab and do a general revision.